to introduce Professor Colin Nichols, whom you all know, and who doesn't need to. But those who don't know, Colin was elected to the Royal Society in the UK in 2014. So I'll just read the nomination statement. I will explain it to you. So it says here, Colin Nichols is distinguished for his contributions to our understanding of cellular excitability and its role in disease. He was instrumental in cloning the first inward rectifier channel and the regulatory subunit of KATP channel. He elucidated the mechanism of inward rectification, generated new insight into lipid regulation of ion channel function, determined the physiological role of cardiac KATP channels, and identified one type of congenital hyperinsulinism. Animal models that he generated predicted the mechanism of human neonatal diabetes and ultimately helped enable patients to switch from insulin injections to oral drug therapy. I think it's pretty good. So, Colin, we're really happy to have you. Thank you for coming.
Thank you.
By the way, those who don't know the history, undergraduate BSc or BA from Leeds, Ph.D. from Leeds, and then a few years of postdoc in John Edwards, obviously. So.
Okay, thank you very much, Jerome. Can you hear me, or do I need to use the mic?
Good.
You okay? All right. So I want to talk about cardiovascular KATP channels, and this is what I have talked about a few times over the last 10 years when I've come and given seminars in CBAC. And I don't recall the last time I came, but I think that the last time I was here, we still did not have what we now know to be the disease caused by gain of function in cardiovascular KATP channels, and that's really what I'm going to focus on here. So in 2 slides, I'm going to give you a summary of everything we know, which is what half my lab has been working on for 20 years, on the molecular basis of KATP channel function. We know that these channels are formed as octameric complexes consisting of 4 Kir6 subunits, each one of which is associated with a sulfonylurea receptor protein. The sulfonylurea receptor protein is a member of the ATP binding cassette superfamily of membrane proteins, which includes CFTR, which generates the cystic fibrosis chloride current, or the chloride current that's lost in cystic fibrosis, also multi-drug resistance proteins. But this combination of a potassium channel, which is a member of the superfamily of cation channels, including all the potassium channels, sodium channels, and calcium channels, shows what I think is a unique pairing, although there have been one-offs in the literature claiming to show association of various cation channels with ATP binding cassette proteins. None of them have really ever been substantiated subsequently. So this is really, I think, a unique pairing. We know from electrocrystallographic studies, which will soon, I'm sure, be beaten by cryo-EM finding high-resolution structures, but electron micrograph reconstructions that we have so far are only at 20 angstrom resolution, and they show only that individual KATP channel complexes are fairly compact, because to fit a molecular model of these 2 subunits derived from related proteins inside them requires a very compact association. So it would require 4 Kir6 subunits in the middle and 4 SUR subunits located around the outside to fill the space available. This cartoon summarizes the basics of biochemical regulation of the channels, and this is what you need to know about to follow the rest of the talk. ATP binds to a site located on the potassium channel subunit itself. These sites are actually at the interface between subunits on the cytoplasmic face, and there are 4 of them for each channel. ATP binding in the absence of magnesium, so it doesn't require hydrolysis. If magnesium is present, you get the same effect, which is to block the channel by causing closure of a gate at the cytoplasmic face of the membrane. In the sulfonylurea receptor, this canonical structure of transmembrane domains, 6 helix transmembrane domains, followed by nucleotide binding folds. There are 2 of these in each SUR subunit. And the way that these 2 so-called nucleotide binding folds generate nucleotide binding sites is kind of like this. If this is one nucleotide binding fold, this is the other nucleotide binding fold, they come together like that. So there's elements from each one that generate one nucleotide binding site here and one here. The 2 sites that are generated appear to, one binds magnesium ATP but doesn't do anything with it, it just sits there. The other one binds magnesium ATP and hydrolyzes it. And when it's in the post-hydrolytic state, I'm not showing you the evidence for this, we could spend a whole seminar talking about this, but when it hydrolyzes to the ADP-bound form, ADP causes some change in the structure which is transduced to the potassium channel subunit and causes opening of this gate, thereby overcoming ATP. So that physiologically, the regulation of the channel is by inhibition by ATP binding to this subunit and activation by ADP, magnesium ADP binding to the nucleotide binding folds. I just want to remind me and you that the sulfonylurea receptors are targets of 2 major classes of drugs, the sulfonylureas and related compounds, which interact with the SUR at some place that we don't really know where, but they cause closure of the channel, probably primarily by interfering with the activating effect of nucleotide interaction here with the channel. Conversely, potassium channel openers lead to channel opening by exacerbating this activating effect from the nucleotide binding folds. Here's the canonical physiological behavior of the channels in an excised inside-out membrane patch which has been pulled from cells which are expressing the relevant subunits. And so this is meant to be an inside-out membrane patch, and so we now are able to apply solutions to the intracellular face of the membrane. And when they're expressing these channels at high density, we see half a nanoamp of current here. Each channel passes about 5 picoamps, so that's about 100 channels open in this patch of membrane. Apply ATP at 1 millimolar when the channels close. 100 micromolar, they're typically about 10% opening. If you add magnesium ADP on top of that, you get a big activation of the channels, and everything's reversible. So, inhibition by ATP, activation by ADP. There's a very interesting genetic makeup of the KATP channel genes. So, we know that on human chromosome 11, there is the gene for SUR1, it's called ABCC, ATP binding cassette family subfamily group C, member 8, is immediately upstream of Kir6.2 K channel family J, number 11. There is no genes in between. There is, on chromosome 12, there is SUR2 immediately upstream of Kir6.1. SUR2 contains 2 major C-terminal spliced exons. The gene contains 2 spliced exons, which gives rise to alternate C-termini to the protein. So we have SUR2A and SUR2B. Somewhere in evolution, then, this tells you first that somewhere in evolution there's a duplication of one pair to make the other one. I think it also tells us that because these genes never got split in evolution, and they go back certainly to fishes, but probably not beyond the vertebrate lineage, but they've stayed together then for millions of years, many millions of years, without the genes separating. So I think that they're actually co-regulated at the genetic level, although we don't really have any evidence for that yet. We know that this pair of proteins are expressed predominantly in the pancreas and neurons in the brain, whereas this pair is expressed predominantly in different muscles. So actually in skeletal muscle, smooth muscle, and also in the heart. And I'll come back to that later on. but I just want to make the point that this pair, we know that they're in these tissues, and when we get mutations in these tissues, actually if we get loss of function in either this protein or this protein, it causes persistent hyperinsulinism because it takes away the KATP channel in the pancreatic beta cell, causes depolarization, constant excitability. The beta cell of the pancreas is excitable, constant elevation of calcium and constant secretion of insulin. Conversely, if we have a gain of function in either this subunit or this one, we get the disease called neonatal diabetes, where we have the K channels are on too much, they're open too much, and that keeps the beta cell unexcitable, keeps it hyperpolarized, stops calcium entry, stops secretion. And in the most severe forms of the disease, when we have very drastic gain of function mutations, and I'll come back to how that is in a minute, we get a neuronal phenotype as well. We get developmental delay and epilepsy. So we've known that for 10 years or more now, and yes, we worked it out in mice first, and we know about it in people, and it's caused a drastic change. It's caused a drastic change in the treatment of those people, and we now know hundreds or thousands of people who've got those diseases. So if we look at a cartoon of Kir6.2 and SURx, this is just to remind me to tell you that there are mutations throughout the channel. Some of them are actually located in the ATP binding site, and these gain of function mutations cause gain of function by stopping ATP from binding to the channel and therefore stop it from closing the channel. Some of them stop the binding, they're not in the ATP binding site, so what they do is they stop the binding being transduced to closure of the channel. Either way, the channels stay open. Some of the mutations in the sulfonylurea receptor, especially those in the first transmembrane domain, which is probably very intimately associated with the channel, have the same effect. They reduce the ability of ATP to inhibit the channel. But some mutations, particularly those located in the second nucleotide binding pore, which is a very big hotspot for these mutations, many of those, instead of changing the ability of ATP to close the channel, they simply increase the ability of ADP to activate it. So they make it more easy to activate it. The net effect in all cases is that you'll get more KP channel than you should have. In the incidence of neonatal diabetes is about 1 in 200,000. In a population of 7 billion, that would mean there's about 35,000 people in the world with this disease. If most of the 60%, most of them, about 60%, have a genetic diagnosis of KP channels, that means about 20,000 people worldwide, maybe about 700 people in the US. And in fact, at Children's Hospital, we get about 1 a year. I want to keep these numbers in mind because I'm going to tell you now about the other disease, and I'm going to argue that the incidence of that is probably about the same. So here's the question. We know that these two genes cause neonatal diabetes. What happens with the other KATP isoforms, which are predominant in the cardiovascular system? What disease do they cause? And just as one illustrative point, Kir6.2 and Kir6.1, these two genes which make up the pore-forming subunits of the channel, are very similar to each other. So the yellow boxes on here mark identity. The red boxes mark some of the early mutations that were identified in neonatal diabetes. You can see that essentially every residue is completely conserved. So those mutations which cause a gain of function in Kir6.2 and therefore cause neonatal diabetes should and probably will occur in Kir6.1 and should cause a gain of function. So having demonstrated 15 years ago with Bob Costa that we could make a deliberate gain of function in KP, and actually we predicted the human disease of neonatal diabetes, we set out to try and do the same thing because, as Yoram is well aware, we've spent 25 years or 30 years not knowing how the KTP in the heart even, what happens when you get changes in KTP function, what they do for the animal. And so we set out to try to understand what would be the effect of increasing Kir6.1 activity. And we did that by making transgenic animals that would express mutations of either this residue or double mutations of both This one's in the ATP binding site, this one's a transduction residue, and each of these residues have been mutated, Q52R and G334D, in neonatal diabetes, and they're very marked gain-of-function mutations. So we made those deliberate mutations in Kir6.1, and we showed in patches, which I'm not showing you, on recombinant patches, that they actually were a gain-of-function of channel activity. Then we made transgenic mice, where we would try to induce the gene in different tissues to find out what it did. And the system we used was one, I forget who used it first, but we started with we got the system from William Coetzee, and we introduced our mutation. And what it does is animals that carry the transgene are green. So these are not dead things. These are baby mice under a UV light. You can see 3 of them that are glowing green because they're expressing GFP, which means they're carrying our transgene. But our transgene won't get expressed until the tissue is crossed in a tissue-specific way with a Cri recombinase expressing animal, which will then take away the GFP expression and express the gene of interest. And so we expressed it in smooth muscle under control of a smooth muscle actin promoter. And these are cultures of smooth muscle, isolated smooth muscle mesenteric myocytes, where they're almost contiguous. Uninduced cultures are very green. All the cells are expressing GFP essentially. When we have induced the animals with tamoxifen, in the same plate of cells, we would see that very few of them are actually green. Most of them have lost the GFP expression. And the consequence of that is that we express the transgene, the gain-of-function transgene, in vascular smooth muscle, and these are just recordings of current at, I think, minus 70 millivolts in, let me get it right, I think it's minus 70 millivolts in, first of all, in 10 millimolar and then 140 millimolar potassium. When we go to 140 millimolar potassium, you see that there is inward K current in control. When we add pinacidil, which is a KTP channel opener, you can see more current coming on, and the currents are strongly blocked by glibenclamide. This is in control. If we look at our transgenic animals, this is one expressing the G343D control. No current at 10 millimolar potassium, but switch to 140. Now we see a bigger current being activated, and even more current being activated by pinacidil. And those currents are sensitive to glibenclamide, although less so than control. And so this is just showing on average, we have more current in high K anyway, and more current activated by pinacidil. So we have introduced a gain of function. Conversely, if we look at cells from animals in which the Kir6.1 subunit has been knocked out, these came from Susumu Sena's lab, you actually see no current under any conditions. So there is no KTP current in those. So it's essentially all through Kir6.1. So what is the consequence of increasing this KTP current with our gain of function mutations? Well, the prediction is that if we turn on a KTP conductance in smooth muscle, we should turn it on in the blood vessels, and that will increase contractility and relax the blood vessels. And what we actually see then, if we measure blood pressure acutely in these animals, is a drop in blood pressure of 10 to 15 millimeters of mercury, both diastolic and systolic. And that's a consequence of reduced contractility. If we look at control mesenteric arteries, looking at, this is pressurized arteries, this was done in Ken Bloomer's lab, pressurized arteries, and look at the ability of phenylephrine to constrict those arteries, it's the phenylephrine sensitivity is reduced about tenfold in the gain of function. So the animals, so the blood vessels are dilated effectively. If we measure the blood pressure in multiple different animals, this is the single mutant or the double mutant that we made. Both of them have lower blood pressure. In wild type animals, 100%, and the control single transgenics don't look any different. If we look at the knockout animals, or if we look at animals using the same system that are expressing a dominant negative subunit that will kill the channel activity, both of those cause 10 to 15 millimeters increase in blood pressure. So what this shows is that with all the normal blood pressure regulatory mechanisms in place, if we change the properties of the KATP channels in the vasculature, we can have a big effect on blood pressure. About 30 millimeters of mercury from having no KATP channel activity to turning it all on. So this tells us that the blood vessels are very sensitive to KATP activity. So we ask the question, are we looking for a vascular smooth muscle disease? So we had this work done, we had the paper written up, and we published it in the Journal of the American Heart Association 2 years ago. But as we were publishing the paper, the real disease appeared in front of us. I'll cut to the punchline and tell you what the disease is first, and then come back and raise questions. So the disease that I'm going to tell you is caused by gain of function of cardiovascular forms of KATP is called Cantu syndrome. Cantu syndrome has a much longer list than this of features, although these are some of the key features that we see. These people have hypertrichosis, that's excess hair on the scalp and on the back and their arms and legs typically. They have hyperflexibility. We've seen people in our clinic that we had here trying to put their middle finger on their forearm. I challenge any of you to do that. They have skeletal abnormalities, which include something called Ale and myoflasc long bones. They tend to have large heads, and they tend to have what have been described as coarse or acromegaloid facial appearances, which includes broad nasal bridges, flat noses, very full lips, and large, broad chins. These people do not have, they don't, as far as I can tell, they really don't have intellectual disability, although there's an interesting psychological point here that is, if you look at somebody who doesn't look normal to you, you tend to think there must be something wrong with them. There must be something wrong mentally. But actually, I met somebody in Holland a couple of months ago with Cantu syndrome, has a Ph.D. in psychiatry, so I think that they, and from talking to them, they really do not have intellectual disability. So there's nothing going on there. They have a huge list of cardiovascular abnormalities, which includes very large hearts. They have increased ventricular mass, and we have now shown that they have, I'll come back to this, they have enhanced systolic function. They frequently suffer from pericardial effusion, which is fluid leaking from the muscle into the epicardium and pericardium and filling up the pericardium, where they have to be surgically drained, and sometimes the pericardium actually has to be stripped from them. They're frequently born with patent ductus arteriosus, which is a vessel that shunts blood across the lungs in utero. They have bicuspid instead of tricuspid aortic valves, pulmonary hypertension, and they frequently have lymphedema. And we see that that is getting worse actually with age, and it can be, for instance, unilateral. Here's one leg that's swollen and not the other one, and I'll come back to that. So Cantu syndrome was recognized by two people who, it would be nice to be here, but I guess they didn't know about it, and I'll come back to that in a minute. The side effects of potassium channel opening drugs, including minoxidil. So most of you are young, many of you are Asian, and may not need to have heard of minoxidil, but some are older and have probably heard about it, and it's sold over the counter as a drug called Rogaine. And you rub it on your hair, and it's supposed to make your hair grow back. And we always knew that minoxidil is a potassium channel opener. We knew that it caused hair growth, but we never knew really why it causes hair growth. Here's a case report from a very old paper, which when you start looking through the literature, you find lots of case reports of overdosing effects of minoxidil and other potassium channel opening drugs, which were under development 40 years ago as antihypertensives, and some of them are still used clinically in acute settings where blood pressure is going through the roof. And if you give these drugs, they lower blood pressure, and they do it through the mechanism that we're talking about. This lady, before beginning minoxidil, looked like that, but 8 months later she had hair all over her scalp, eyebrows that were meeting in the middle. Her face looks more swollen and maybe edematous. She's got these deep creases here on the side of her face. You wouldn't recognize her as the same one. And this is just a point about this Rogaine minoxidil. They call it Regaine for women. And this is what it can do to some lucky fellows. But it can cause all these same side effects, including lymphedema. And if you look in the drug companies' literature, it doesn't tell you how the minoxidil causes hair growth. But we know now that people who have Cantu syndrome, caused by gain of function of KATP channels, as I'll tell you, have got the same features. So this was first recognized, the similarity between the drugs and the disease, by Kathy Grange and Gautam Singh, who were in the Department of Pediatrics at Washington University School of Medicine in St. Louis, Missouri. And they wrote in this paper 10 years ago, looking at a woman, and I'll come back to this woman and her two daughters. They were showing through this paper that it could be inherited as a syndrome. Nobody knew at this point how it was called, and I'll tell you that 3 years ago there were 12 papers in the literature on Cantu syndrome. So extremely rare, completely not studied by almost anybody, and not known by essentially any clinicians. But what they wrote in this paper was that because the effects are similar to those of the side effects of minoxidil, which acts on K channels, it's possible that Cantu syndrome is caused by a defect in a K channel gene that decreases vascular resistance. And they had a colleague in the division called Kir6.2, who I knew, and who told them at the time, oh, there's a guy in the medical school who works on those channels, you should go and talk to him. 10 years ago, we could have figured this out. If they'd showed me all of this, I would have said, yes, this has to be KATP. We could have done the sequencing and found it, because we'd already missed out on neonatal diabetes, finding it in people. But they didn't, and actually, that's why I keep giving this talk, a related talk. I don't know if there are any clinicians in the room, I doubt it. Are you? You got a time, you probably are, okay. Well, but I've given this, essentially this, I gave it in Vancouver, and I gave it in Columbia, Missouri in the last 2 weeks, not one in the room. And my point is, you know, if we talk to each other, we can learn things. So, these two papers appeared in Nature Genetics and American Journal of Human Genetics, which, for those of you who are not geneticists, are top journals, 3 years ago. Both of them describing how coding mutations in ABCC9, which is the SUR2 gene, cause Cantu syndrome. Between the two papers, they found coding mutations in 25 out of 31 known patients with Cantu syndrome. We since now, between us and them, and we're working with them, have identified maybe 100 people, but going back to what I said in the first place, there are going to be tens of thousands. If you look at these, you'll note that these people all have Dutch names. And well, many of them are Dutch, and I thought, well, that's kind of weird, two papers both from Holland. But I did notice that the third author on this paper was somebody called Dorothy K. Grange, and I thought that sounds like a very English name. And I looked up where she was. She's at Washington University. So it took me about 48 minutes from reading this paper to finding her, and then, we've done a lot together since then. So SUR2 gain of function causes Cantu syndrome. Do we know it's a gain of function? Well, the only evidence we got from those first papers was that if they express this SUR2 mutations with Kir6.2, they actually did, for technical reasons which I'll come back to, they did see that there's a gain of function in the generated channels, and they are less sensitive to ATP than wild type. So these recordings of current for wild type and 3 different mutations, they're showing ramp currents from minus 100 to plus 100 millivolts in 140 potassium, so they reverse at 0. These channels are really not inwardly rectifying, they're almost a linear K conductance. Under control conditions, there's a very weak inward rectification, but as you add ATP, and in this case they're adding magnesium ATP, so they're conflating both the inhibition effect of ATP and the activating effect of magnesium nucleotides at the nucleotide binding folds. But you can appreciate that if you go from control to 0.1 millimolar, 100 micromolar here, there's about 50% inhibition, whereas, and then you go to 10 millimolar and there's complete inhibition, whereas if you look at each of these mutations, they're much less sensitive to adding those ATP concentrations, and that's borne out on the right, where this is at minus 100, this is at plus 100, it's redundant, you're looking at the same thing, just looking at the currents at minus 100 or plus 100, you see that there's a rightward shift in the apparent ATP sensitivity of the channels. And this is basically the same kinds of rightward shift that we see causing neonatal diabetes when we look at Kir6.2 and SUR1 mutations. This is a model generated by Sanju Lee showing both the open and closed conformations of the SUR subunits, bacterial homologs coming to open and closed conformations. These are the transmembrane regions. These are the two nucleotide binding folds, and this is the interface between them. You can see that these mutations that are in Cantu syndrome are dotted throughout the subunit, but a couple of them appear in the middle of the nucleotide binding folds where the interface is between the two nucleotide binding folds. And in a systematic study of the predicted interface in SUR1 that Ricard Macia did, which was a big part of his Ph.D. thesis 10 years ago, he identified most mutations in the interface would actually decrease SUR function and decrease ADP activation of the channel. But right there in the middle of the interface, you could find some mutations which actually seem to lock the dimer together and enhance activation. So I think that's part of the mechanism in this particular case. So I said that, I was careful to say that in that paper they looked at the effects of putting SUR2 with Kir6.2 to generate the channel. Now, if mutations in Kir6.2, as I've said, cause neonatal diabetes, they do not cause Cantu syndrome. And thus far, we see no overlap of the symptoms of neonatal diabetes, or DEN, the epilepsy and developmental delay, syndromic version of the disease, and Cantu syndrome. So Kir6.2 cannot be generating the channel with SUR2. So the logical subunit that would have to do that would be Kir6.1, for which we know of no disease, but that's the other canonical partner. So it should be that Kir6.1 will cause Cantu syndrome by the same mechanism. I said that those first two papers identified 25 people out of 31 with the disease. The other 6 did not have ABCC 9 mutations, but one of them actually had a mutation in Kir6.1. And it turned out that that little boy was a little boy of Kurdish origin in Germany, hence the reason that these are actually Germans, not Dutch. And that's the little boy I showed you on one of those earlier pictures. And it turns out that he has a mutation. This is him now. By the way, that paper I just showed you was in 2002. Now he's about 15. He has a mutation, cysteine 176 to serine, that is right in the middle of the second transmembrane helix of the predicted structure. And it turns out that the equivalent residue in Kir6.2 is C166, and C166S is probably the most studied mutation in Kir6.2, and it causes a very marked gain of function by locking the channel in an open configuration. So we looked at this mutation in Kir6.1. I mentioned that for technical reasons it's not easy to look at Kir6.1. The channels disappear in patches. It's very hard to find them. But what Paige Cooper, who just graduated with her Ph.D., figured out was that actually by mixing Kir6.1 and Kir6.2 to make heteromeric channels, we could get robust currents that were stable. And with, I'm not showing you all the average data, but just some raw traces. When you mix the two, you get high sensitivity to inhibition by ATP in wild-type mixtures, but if you mix the C176S mutation in Kir6.2, you can see very ATP-insensitive channels, which you never see them like this for 6.2 alone. So it's demonstrating that, in fact, we do have a gain of function from Kir6.1 and SUR2 in this case. You can see that this boy, even though he's now, in this picture, I think he's about 13, but from behind he looks like he's probably 6. And he still has a very large head for a boy of that age, but you can see he's still got a lot of hair on it. He has this massive pompadour covering his hair that's on his forehead. And I think this appearance of looking very young, we've seen this now in many other people, and I think at least until puberty, they tend to look very young. Okay, so I think this all shows that SUR2 and Kir6.1 are causing Cantu syndrome, but how is not clear. I gave you that long list of features, and I challenge any of you to give me a good explanation for more than one of them from a gain of a K-channel function. So the first question is how, and then secondly, what can we do to help these people? So what we've been working on with Kathy and Gautam and a whole group of people now is setting up a program that goes from patients to animal models to cellular models, and then hopefully we are looking to go back to therapeutic trials of drugs that will actually address these challenges. So we set up this a website and a registry for people to find out about the channel, which we run from a program here. We now have a REDCap registry of patients, which is including the people from Holland, and I will say that we now have about 100 or more people who have been diagnosed from around the world who are on the registry. And by diagnosis, typically that means genetic, but not in every case has it been confirmed, but most of them have been confirmed with mutations in, most of the mutations are still in SR2. We know of 2 mutations in 61. We've signed on many families, and we've set up an annual clinic here in St. Louis, which we've held for 3 years, and they held the first clinic to do the same thing in Holland this September. And this actually is a photograph from the first clinic that we held here in St. Louis, and this is the mother and 2 daughters that were described in that 2006 paper from Gautam Singh and Kathy Grange. And these are 2 other, well, I say children. This girl is, I think she is 18, and this boy is 13 from that first clinic. Sorry, I'm trying. So you can see actually they all look young. The 2 daughters here, this one is, I think she's 21 in this picture, and this one is 18. But for those of you who've got teenage daughters, you know that they can look a lot older than that at those ages. So there's this long list, I said, this long list of features, and the question is, where are they all coming from? There's no mention, by the way, of QT shortening. So the first prediction of having a gain of a K-channel function in the heart, one that's a linear K conductance, it's not even an inward rectifier, it should shorten the QT interval. It should reduce the cardiac output, but these people have enhanced cardiac output. They've got big hearts with enhanced systolic function. And there's no mention of low blood pressure. This is the one feature that we predicted directly from the mice. Why not? So are we really looking for a vascular disease, as I had originally said? So one of the first things we did in the clinic, and Mark Levin was instrumental in getting all the data together in the first place, was just to measure carefully the blood pressure from these people when they were sitting down and when they were standing up. First point to note is that clinically they don't faint when they stand up. They maintain their blood pressure, so every clinician will tell you they've got normal blood pressure because they don't faint. Not, as a physiologist might say, because their blood pressure is 120 over 80. No, and in fact if you look at their blood pressures, their actual numbers are much lower than you would typically see for anybody of their age. So these are the actual ages of each of the patients. And this is the age at which their systolic blood pressure would represent the 50th percentile. So just to pick one example, here's a 12-year-old boy with the blood pressure of a baby, and a 23-year-old with the blood pressure of a 6-year-old. This woman who's 47 and has normal considered blood pressure has been under all sorts of iatrogenic insults, for want of a better word. She was being treated with lots of medications, including calcium channel blockers at one time, which are probably completely counterintuitive once you know it's a gain of K-channel function. So, in fact, they do have low, physiologically low blood pressure. And they do have vascular dilation. So these are MRIs of the head that were done by Jin-Moo Lee and collaborators in neurology. And it looks to me, although I'm not a neurologist, so I can't really say, it looks evident to me that their blood vessels, and I'm told by them that in fact their blood vessels are, these are the cerebral arteries, are dilated, and moreover they're tortuous, they're extended. So there is tortuosity of the vessels. In other words, they're longer than they should be. And moreover, what I'm told, they can detect the presence of persistent fetal arteries. This is a trigeminal artery, which normally disappears after birth, but it's still present here. And in MRI of the thorax, Gautam Singh assures me that there are arteriovenous collaterals which should not be there. So I think actually we're beginning to see part of the explanation for the persistent ductus arteriosus. Not only do they have a persistent ductus arteriosus, they probably have persistent fetal circulation all over the place, because there is this pruning of blood vessels that occurs after birth, where the vessels constrict, close off their own blood supply, and thereby die and turn into ligaments. And if you have a weakened vascular contractility, it fails to close, stays open, stays patent. What's going on in the heart? I may not have time to go through it all, but I'll come back to this one point. The density of KATP in the heart is, in most animals, enormous. And this is from rat. This was our calculations. It's bigger than the sodium current. It can be enough to shorten the action potential to nothing and even stop the heart. But the ventricular KATP channel is mostly closed because channels are inhibited at the existing ATP concentrations. And because of this, in animals, if you knock out KATP channels or you put on KATP channel blockers, they don't do too much in the normal heart. That's why it's possible to go through a whole career in cardiac electrophysiology and essentially ignore the KATP channel. But anybody who actually tries to look at it, either pharmacologically or activating it by metabolic inhibition, sees that there's this giant conductance sitting there, as Yoram knows. And so if you put a gain of function in, you can have a massive effect. And these are actual recordings of rat action potentials in metabolic inhibition, showing the action potential shortening at different times. And this is from a calculation that I did about 100 years ago, using Dennis Noble's Oxsoft heart model, predicting how the action potential would change if you activated this KATP conductance as ATP dropped. It would have to drop quite a lot. And Yoram and what was his first name? I can't remember. Robin Shaw did the same kind of calculations, came up with the same prediction, which is that actually, even with normal ATP sensitivity, you only have to activate about 1% of the total KATP conductance to shorten the action potential by 50%. So the action potential in the heart should be very sensitive to activation of KATP. And if you get ATP-insensitive channels, then you should be able to really shorten the action potential. So what is going on in the Cantu heart? Well, these are some recordings that Mark and co did from Cantu patients. Short answer is actually not very much. There is no evidence for QT shortening in any of them. There's a little bit of PR prolongation in some of them, and there is some evidence for T-wave inversions going on and flattening of T-waves. So not much, and no overt arrhythmias. If we look at our animals in which we've deliberately expressed gain of function in the heart, we can see AV dysfunction, so we can see where we will get a block, so we can see the SA node going on at its own rate, but we see the ventricular rate is beating it out. So we do see evidence of AV dysfunction, but in the Cantu patients, no evidence for QT shortening, and if anything, flattening and inversion of T-waves. And I'll come back to what that could be in a minute. We can show in the patients by looking systematically at Cantu patients that they really do have bigger hearts. Left ventricular end diastolic volume is increased. Left ventricular mass is about double in these people. The extent of shortening is greater, so the cardiac output is greater, so they have enhanced contractility. We made these different kinds of transgenes. In the first place, what Bo Costa did 15 years ago was make KATP Kir6.2 gain of function mutations, where he would express either a single or a double mutant. It doesn't really matter what the mutations are, but it puts in a gain of function into the heart. And what we showed in those animals was that the ECGs are essentially normal from those animals. There is, if anything, a very slight slowing of the heart rate, maybe a 10% slowing. And Tom Flagg did careful analysis of the currents in myocytes from those animals The KV currents were really not affected at all, but there was some evidence of a slight increase in a background K conductance. If you look at the outward currents and you look at the steady state current density, this is from control, and this is from the gain of function animals, or predicted gain of function animals. You see this is very weak. It's slightly inwardly rectifying, it's reversing at some negative potential around -50, but it's getting into the noise here. So some evidence for a very small activation of a KTP current. But the most dramatic thing we saw was a huge increase in the L-type calcium current. Putting a gain of KTP channel function in the heart gave us a doubling, almost, of the L-type calcium current. So these are families of currents from wild type and mutant. This is wild type in control. This is line 4, which was a high expressing animal, and the line 2, which is a lower expressing animal. So bigger L-type calcium currents associated with bigger enhanced contractility of isolated myocytes. So just measuring the amplitude of contractions, isotonic contractions in isolated myocytes, 10% shortening at different stimulus frequencies in control, about half as big again in the transgenics. And if we look at the calcium dependence of contractility, it's left shifted. So we've got enhanced contractility because of enhanced intracellular calcium, because we've got these bigger calcium currents. And what we found was that even though in control, anybody who works on calcium currents, if you put on isoproterenol, you typically get 50% to double the L-type calcium current. But in these transgenic animals, we didn't get any increase with isoproterenol. So they're pre-stimulated somehow. And we've now repeated this in an extensive study that Mark has done using Kir6.1 gain of function. So now a completely different transgene in which we introduce Kir6.1 into the heart. And we see essentially the same phenomenon, which is increased ejection fraction, increased strain rate on echo in the hearts themselves. In the isolated myocytes, we see an increase in the baseline contractility, but no increase in isoproterenol. And if we look at the calcium currents, we see the same thing. So control and the gain of function animals, we see about double the calcium current. And if we add isoproterenol, they're both normalized. So the same effect from a totally different transgene, but it still introduces a gain of K-channel function into the heart. And with the help of Bill Cattrall, looking at phosphorylation of these subunits, we see that in the cardiac gain of function, not sure what I'm doing with this, but the red one, if you look in this, okay, there. The red one, if we look at phosphorylation of one particular residue, which is actually a controversial residue, it may be phosphorylated by pKa or pKc. We see a big increase in phosphorylation in our gain of function animals. And I haven't gone on too much about this, but if we express the gain of function in the blood vessels, which is the one that gives the dilation of the blood vessels, we also see a smaller increase in phosphorylation of this residue compared to the relevant control. So what does all this mean? In a complicated way, potentially, if we have gain of KTP channel function in the heart or in the vasculature, the primary effect of either one of those should be to decrease cardiac output by decreasing, or in this arm, by decreasing heart rate or action potential duration. Both of those will decrease calcium, decrease contractility, which will tend to decrease cardiac output, which would therefore tend to lower the blood pressure. If we have overactivity in the blood vessels, decreased electrical activity, decreased smooth muscle calcium, decreased contractility, decreasing peripheral resistance, again lowering blood pressure. These effects will then give central drives to stimulate the heart. Either one of those will tend to cause neurohumoral signaling onto the heart, which we don't know exactly what's going on. We don't have a good way to track it, although we do have some evidence that there is decreased heart rate variability, both in Cantu people and in animals, which is consistent with alterations in autonomic activity. And it appears that we have a marker, at least in this serine 1928, which is increasing the L-type calcium channel activity, at least in the heart, which will tend to counter these negative effects. So what we've been looking at all along is a massive feedback secondary consequence that's actually maintaining what would otherwise be a much worse lowering of cardiac output and blood pressure. So in conclusion, I mentioned briefly that if you have gain-of-function mutations in either of these subunits, you get neonatal diabetes and Den syndrome. It's replicated in animal models and can be treated by, I didn't tell you this, but because I cut this out, but it can be treated by sulfonylurea drugs, and sulfonylurea drugs have proven to be a magic bullet treatment for these people. They have stopped taking insulin, they take sulfonylureas once a day, and they're well controlled. So now I showed you that these two genes actually cause Cantu syndrome, which we can also see replicated features in animal models, and we're beginning to explain some of the complexity. Can it be treated by sulfonylureas or other related KATP channel blockers? I didn't show you the data, but we've made specific CRISPR-based knock-in animals that are also starting to reiterate the key features, and we are beginning to look at the different drugs that will be able to do it. So hopefully next time I'll tell you that we have succeeded in correcting the problem for these people. So there's a lot of opportunity to generate a new drug. Oh, a new drug for Cantu, which is already on the market. No, but it turns out that there is this stuff in the supermarket, Cantu products, which give you beautiful natural hair. I cannot believe that this occurred randomly. Apparently, this is used by African Americans predominantly. So when I showed it to Paige, who is African American, I think she was a little taken aback that I thought this was amusing to see it in the supermarket, because to her it's probably like looking at Purcell or something. She's used to seeing it all the time. Anyway, I think so. There are a lot of drugs available. I've acknowledged many of the people who did a lot of the work, but I think it's worth saying that much of the work on Cantu was done by Tom Flagg, Aisha Zhang, Maria Remedy helped making the animals. Amir Toy did some of the work. Bo Costa was responsible for initiating all of it. This is John Silver when he was a young boy. And I also owe a debt of gratitude to the clinicians who helped us. This is the chap who did the very first sequencing on a Cantu patient in Holland, and he's the one running the Cantu group over there, Kees van Haften. This is the mother and two children. That's Gautam Singh, that's Kathy Grange, and that's Mark Levin on his knees. Okay, thank you very much. I'll stop there.
Thank you, Colin, for a fascinating story. So what happens to a Cantu patient if you're too young to know, but what happens to them if they have an episode of acute ischemia?
I don't know.
What happens to your animals if you close it to them?
So we haven't yet done anything on ischemic challenges or blood vessel.
I'm talking about, the inability to shorten ABD.
Yeah. So there is a big prediction that they will have an inability to counter-regulate. That's one thing in the blood vessels. What happens acutely in the heart, we don't know. I do know that if you knock out SUR1 or SUR2, those are supposed to be cardioprotective in ischemic models. I don't know why.
Very interesting to look.
Yep. We do have some evidence for potentially less exercise tolerance in Cantu patients.
So it's maybe a common feature of ATP channels. Can you hear me?
I can hear you. I didn't hear what you said though.
Okay.
No, I can hear you. Just say it again.
Okay. So I'm saying, ATP channels are can be activated by ADP.
Yeah.
Right. So and there is no doubt it's not phosphorylation effect. It's ADP binding, right?
Yes. It's ATP binding and it's ATP hydrolysis to ADP that is giving the activating effect.
Okay. So can this channel be activated by AMP? Monophosphate.
It's very weakly inhibited by AMP. No evidence for it being activated. There is some data on AM. What's the compound? There's a more complicated.
So actually, this second phosphate is important for activation, right? It seems so. It's not nucleus.
I think what's important is hydrolysis. So most of these ATP binding cassette proteins pump things by ATP hydrolysis. So I think it's necessary that the nucleotide binding pole cycles.
Yeah, you mentioned that actually two nucleotide binding sites are next to each other, right?
Yes.
Adjacent. So can it be activated and activated by phosphate transfer from?
So there is a question that was looked at somewhat in CFTR about phosphotransfer between the two NBFs. I tried to repeat those kinds of experiments in KTP and couldn't see anything, so I wasn't convinced by any of that actually.
So you have a gain of function that causes disease, but you don't have any loss of function. So is 6.1 a vestigial channel? Like it just kind of stayed along for the
I don't
ride?
think it is. I think we don't know yet the real human loss of function disease. It's the anti-Cantu disease. It has to be some sort of hypertensive phenotype. And people have looked for association of 6.1 variants with hypertension unsuccessfully. The only papers I know where people have claimed to show effects of loss of function mutations in human disease have been for SUR2. There's a couple of reports from Andre Terzik's group of, I think it's congestive heart failure associated with SUR2 loss of function, but they're one-offs, and whether it's a causal link, I don't know. There was also a paper with Ackerman as a senior author on several cases of SIDS babies who had reported loss of function mutation. They reported loss of function mutation in Kir6.1, but any causal association to the disease is unknown. So I think it's some sort of, so the animal phenotype mimics Prince-Metal angina. So you get a hypertensive phenotype and you get spontaneous coronary vasospasm. So, but people who have Prince-Metal angina have not been found to have loss of function mutations. So again, we don't really know. But it's still out there, and there's going to be, I'm sure there's going to be a clear association somewhere, but we don't know what it is yet. But it's worth keep looking.
I was wondering why, does anybody know kind of why there's such a high density of the KATP channels if they're not, if not very many are open?
Yeah, good question. So teleologically, why are they there? This same question we and others have been thinking about for 30 years. It actually opens up, so I think in the vasculature they're dynamically operating, and they're dynamically operating to control blood pressure. Just as in the pancreas, we know that KATP channels are dynamically operating to control insulin secretion. In the heart, they don't seem to be. And what I didn't really draw attention to is that in the heart, one of the major pairings that we see between Kir6 and SUR is the non-canonical pairing of Kir6.2 with SUR2. So the genes are coming from this pair and this pair. So it's not the canonical pairing. So I have a feeling that the heart, and this comes back to Yoram's point, the heart is essentially forcing, how it does it or what, I don't know, but it's forcing a non-canonical pairing of these subunits so that they're not dynamically active, so that under normal conditions you have very little activity of these channels. But they act as a kind of safety valve that comes on only under drastic conditions. So what you see, if you look at isolated myocytes or whole heart, is KATP is not a continuum. It's really not there. It's closed, so you're not seeing much. But when it comes on, it's huge. So I've always had this notion that what may be happening is that it may come on in very small regions, but you don't see it macroscopically. So if you look at the electrocardiogram, you don't see it because the length constant for myocytes in the ventricular myocardium is probably 2 or 3 cells, would you say? Probably, let's say 2 or 3 cells. And let's say the capillary beds are such that as it gets down to the finest capillary beds, there is almost 1 vessel per cell as you go through the myocardium. So let's say, and you may have seen these videos of blood flow through the smallest capillary beds where you see cells going like this, and then one will jam like that. And it'll jam in the wall, and it'll just sit there, and there'll be no flow. And then at some point, it goes again, but it can sit there for maybe minutes. So if you have a blockade in a very, very small capillary, and it stops the blood flow to a small, very small region, say half a dozen cells, then that might be enough for their metabolism to drop as they exhaust the oxygen in them and generate CO2. Might be enough for them to suffer damage, but the KATP comes on and stops the electrical activity in that tiny region, stops them from contracting. The bulk myocardium is still doing everything, but it's protecting that little region and maybe protecting it enough until flow starts again, but you don't really resolve that macroscopically. That's kind of the only argument I've been able to come up with, but it is interesting that we have this non-canonical pairing, and we have no overlap of Cantu and DEND features, so really, that we know about. So what is really going on for gain of Kir6.2 versus SUR2 in the ventricular myocardium, we still don't know, and it comes back to your point that it might all have to do with more drastic.
Well, the other point, just to continue on this, is, it takes, quantitatively, it takes to open 1% of the channel density to shorten action potential duration by 50%. It's hugely non-linear.
Yeah.
So, because it's all stochastic, you want to have that kind of reserve built in with high density, but you really want to operate only on a small part of the available density.
Yeah.
Because of the stochasticity of all of this.
Yeah.
So that's, I think, that's the idea.
Yeah. Yeah, there is a huge redundancy effectively in the heart because you can really stop it if you activate enough of these channels.
Yeah, question. Have you observed the resting potential of the mouse gain of function model? The resting potential depolarized or hypolarized?
When we looked 10 years ago, Tom Flagg at the Kir6.2 gain of function,
Yeah.
there was no, to the best of it we could see, there's no change in the resting potential. It still sits at -7.
If there's no overlap with the gain of function in SUR2 and with the heart conditions and DEND, is there a converse effect of the hyperinsulinism? Do people with loss of function in SUR2 have cardiac phenotypes?
People with loss of function in SUR1, you mean? Do they have a cardiac phenotype? In hyperinsulinism?
Sorry.
Or Kir6.2?
6.2 is what I meant. not
Yeah.
SUR2.
No, nothing that I know about. So I suspect that in the end, Kir6.2 gain of function does have some effect in the heart that is also there for SUR2. But it's dragging out what the condition is where we really see that we don't know yet. We have to go to experiments where we're doing, coronary occlusions and maybe afterload increases on the heart. Probably what we'll do is we'll get into heart failure in the end.
It'll be interesting to see if you include.
Yeah. But we don't know yet.
Okay, any other? Okay, Colin, thank you so much. And thank you.